project today's workshop on the African BioGenome Open Institute for Genomics and Bioinformatics workshop on genomic infrastructure, biodiversity genomic infrastructure, and yeah. So today we are going to start right away because we are on time, and we are going to be recording live stream, streaming this live, and as well as allowing others who have not registered to join us on the other AfricaBP platforms. So once again, I'd like to especially welcome you to this first day of the workshop. So today we have our a program which is already been communicated to you via email. And I believe that all of you have read and understood how we are going to proceed with this workshop and for these 2 days. So yeah, for a go with the first presentation, I would like to welcome Charlotte, who is going to be giving us a talk on the African BioGenome Open Institute. So Charlotte, I'd like you to come on the platform and give your presentation. Is Charlotte in the call? Hello, Charlotte, are you on the call? Okay, Charlotte is joining us. Charlotte is joining us. Just oh, just a moment, just a minute. Then Charlotte is joining.
Taiwo, I think let's move to the next presenter, please. Charlotte is not here, so we can.
Oh yeah, I thought that Charlotte was logging in. Okay, so while Charlotte is joining the call today, but for then, we would like to just proceed to welcome for our presentation, Alice Matiba. Alice, are you on the call? So Alice will be talking on training and capacity building strategies for global genomic training. So Alice, are you on the call? We would like you to make your presentation on that. Alice? Great. So please, you can share your PowerPoint. You have been made a co-host. So Oh yeah. Thank God, can you unmute Alice?
Yeah, you can actually unmute her, Taiwo. You have
Okay.
why a co-host here.
Yeah.
So just to mention that all participants are automatically on mute, so only the co-hosts would be able to unmute.
Okay.
And this is basically because of the number of people attending, so we can be able to control the audio.
Sure. So Alice, you're now mute. So
okay, can you all hear me? Can you hear me, Taiwo?
Yes, I can hear you very well.
Okay, great. Just checking. Okay, I will share my screen now.
Great.
Slides. So hello, everyone. I'm happy to be presenting to you today, and thanks to the organizers for the invitation. I'm Alice Matimba. I'm based at the Wellcome Connecting Science, which is based at the Wellcome Genome Campus in Hingston. And I will be talking to you about some of the training and capacity building strategies that we are implementing on our learning and training program. So some of you are already quite familiar with our program, and I was going to ask, if you can raise your hand, but I probably can't see everyone on my screen at the moment. But so basically, we are based at the Wellcome Genome Campus. We work closely with the Wellcome Sanger Institute and also with EBI on the campus that you can see a picture there. So today I'm going to introduce you to some of the work that we do on our courses and global training program. And then I'll go into some of the top level strategies that we're implementing. And then finally summarize with a framework for what I think would be useful for the Africa Biogenome consortium to apply in terms of developing training for their audiences. So okay, so Wellcome Connecting Science, as I mentioned, is based at the Wellcome Genome Campus, and there are two divisions. So the Learning and Training Program, which is where I'm based and leading the courses and global training program. And then the Engagement and Society, where the team are focusing a lot on research and ethics, but also doing a lot of public engagement activities, and also some training of public and high school children. So the Learning and Training Program it develops training for building capacity in genomics. And this is working through partnership and collaboration with UK scientists and globally as well. We also are contributing to development of genomic medicine through provision of education and training to clinicians and healthcare professionals. Obviously, we work with other, organizations and other upcoming communities within the genomics field as well. So we mostly run a lot of the courses previously, obviously before the pandemic, we're running a lot of the courses and in person based at Hingston, but also in some locations in Africa, Asia, and Latin America. My colleagues, Tressa is heading the conferences team and also the online courses with Rachel Beckson. So we usually are, aim to run about 60 events a year. So this includes the courses and the conferences as well, both here in Hingston and also in the global regions. So in the learning and in the courses and global training program, we are obviously, spend most of the focus is around genomic skills building and training. And this is through laboratory-based training, but also genomic analysis and bioinformatics. We also have some activities to do some research and also to development, particularly around how we can, developing strategies or formats that can help us to have more impact globally. And I'll talk about this shortly. And then we also have an area which we also think is quite important in capacity building, which is in professional skills development. So here's the QR code to our webpage. So you can, just turn on your camera and you can have a look at our website if you want to see more or look at all the different types of training that we're doing at the Wellcome Connecting Science. But basically, our courses are divided into, different applications and themes. So we have courses that are focusing, for example, on pathogen genomics. We have also training that's focused more on human genomics. We also have training that's focused on, application of technologies. And particularly in the main focus has largely been for clinical and healthcare applications and disease surveillance. So if you could take a look when you get a chance, and we do have quite a number of courses that are open and conferences as well. And the courses that we have that run in the global training program, for example, are completely subsidized and free to attend. The courses that run here are also open to anyone and everyone globally, and you can always also apply for bursaries. The conferences also run events that are sometimes in virtual format or also in hybrid. And again, you're welcome to apply or submit an abstract, and you can also apply for bursaries, particularly if you're based in low and middle income countries. So we run a series of, we've developed quite a number of delivery formats. And obviously our, the main format that everybody likes to have is the face-to-face training, which are based in Kingston and abroad as well. And usually these are running for 5 to 10 days. We also sometimes collaborate with conferences or consortia that are running meetings on hosting 2 to 3 day workshops, particularly in a targeted format to conference attendees. And due to the pandemic, and I think just like everybody else, we transitioned into virtual delivery. And this was so a very successful transition into virtual delivery. And we ran over 35, and I think there's probably more virtual courses. And also in the conferences side, also I had ran over 40 virtual conferences with thousands of participants being reached globally. And so we have perfected the art of virtual delivery. And although we've perfected that, we are now actually moving back to in-person events. But we hope that we can continue to offer virtual delivery for some of the training as well. We have also been collaborating with the H3 Africa Bioinformatics Network on distributed classrooms, and I'll mention this a little bit later, and this is usually running over several weeks. We have an online courses program, so this is running off the FutureLearn platform, and I think some of the scientists that are on this call are collaborating with us on a course for which will be targeted for the similar audience that is on this call. So hopefully you'll get to see some of the collaborative work that we're doing with consortia such as the African BioGenome. And then of course we also have a podcast, and I will mention that later. So in our organization, these are some of the sort of like special, things that we have to offer our audiences is that we actually have a dedicated full-time team that are responsible for working with experts to develop content. We have people specializing in IT and lab, and also the event organization and marketing teams. So this allows us to reach more people. This allows us to have a full-on schedule running throughout the year, and also keeps us, keeps us quite busy, but also connected with other communities as well. Our training, as I mentioned earlier on, is subsidized, both on the global training program, but also in Kingston. And some of the courses and conferences are free to attend, particularly for people based in Africa, Asia, Latin America regions. Key to all this, and we don't do this alone, and this is a very important part of our of our program, is that we collaborate with global experts, and most of the work that we do is attributed to the scientists that we work with. And so we take it quite seriously to go out there and develop partners and partnerships with organizations which help us to be able to expand the reach in different regions, but also to make sure that we're tailoring the delivery to the needs of the different audiences that are also, again, quite diverse in different regions and so forth. So our success is actually attributed to the collaborative partnerships that we have developed over time, and we're continuously looking to see how can we do better with these different groups. So now I'm going to move on to some of the work we do in the research and development. And this obviously is including, we obviously are researching to understand training needs and evaluating impact. We also use a lot of this information to develop new content, for example, and we're constantly reviewing the program to see how it's aligning to interests in the, in the ever fast moving environment of genomics, and also the changing needs of the different communities. We also conduct some research which enables us to improve or improve the quality of our program, and also increase the capacity. So here's some of the feedback that we get from participants. So this is feedback that's coming from participants that were on the global training program. And we are always quite pleased to see that most of the people are actually going back and applying what they've learned. And most of the feedback, and we do open up to people providing us with more information about what they've done when they've gone back. And we're also opening up discussions around how we can support people to be able to share or apply what they've learned. And quite a number, and some of our programs are also trying to encourage participants to pass on what they've learned to other scientists, especially in their own home environment, just because for most times we're only able to take on a limited number of people on our program, and therefore a key requirement is that they actually go back and teach others and train others. And in order to do so, we do have some resources that we provide our participants with. Okay, so we also are conducting some needs surveys. So most of our program, we, especially on the global training, we try to not, put everybody in the same blanket, because, the different regions have different needs and or interests. And so we've been conducting a series of training needs surveys and analyzing our data based on the regions, but also trying to understand the needs for researchers, for example, versus those of healthcare professionals, and also trying to understand what do they need now, but also preparing for what do they need in the future. So we've been gathering some data, some information through surveys, but also talking to other scientists in the regions, and we're open to hearing more from you as well. And I will share a survey at the end of this meeting. So in terms of capacity development, Our goal is to improve access. So we also want to, expand reach so that we're reaching more countries, and but also to always be thinking about sustainability issues, because we know that there isn't a bottomless pit of funding, for example, or resources. So sustainability issues have to be factored in to our program, but also with the people that we are collaborating with. And it ultimately, that all this increases the impact that we want to see. So, so in terms of trying to expand reach and scaling up, we have partnered up with the Africa Bioinformatics Network to expand one of the the training in one of the most popular courses that we run, which is the MGS Bioinformatics course, where we usually receive a ratio of 4:1 applicants versus selected participants. So you can imagine that a lot of our courses would be taking about 20 to 30 participants at a time. And by using this model, we've been able to reach, very, very high numbers in the hundreds at a time, located in different regions in Africa, and it has been quite successful. And so having piloted it in 2021, we've run it again in 2022, where we had about 40 classrooms and reaching over 600 participants who were selected, and also empowering some of the local scientists and who were part of the classroom staff. So another area where we've been doing a lot of research and experimenting is in the train the trainer, using train the trainer methods in order to build capacity or to expand that capacity. And this is through providing some of our participants who come on our short courses with some resources on to help them understand how they're learning, but also how they can go back and train others. So this is usually a very short module where we provide them with the tools to actually even just design or plan how they're going to train others, how they're going to present to others, or in the form of, it could be presentations, it could be departmental presentations, it could even be a report. So we try to encourage that so that participants are not just coming to learn the genomic skills, but they're also learning how to pass it on, how to share knowledge with others. We also have dedicated train the trainer courses, which we've run in the face-to-face formats and also online using the FutureLearn MOOC platform. And also we're planning a virtual course, which I'll mention again a bit later. But we also take advantage of using, working with, we work with experts, we collaborate with, experts and scientists globally. And so we provide opportunities for, say, early or mid-career scientists that are interested in developing training skills to be part of training teams, and they can get mentored by some of the experts that are already quite experienced in training. So there are different ways that we have been developing the train the trainer strategies, and we've had a couple of presentations about this, where we just explaining the different models that we have been applying. We've also been collaborating with other programs that have been who have been focusing on establishing capacity in global regions, with the particularly around genomic surveillance of AMR, and working with a group with the Center for Genomic Pathogen Surveillance. And of course, as I mentioned earlier on, we've got an online course that is been developed also, trained to design, to help put scientists or anyone interested in designing genomics and bioinformatics training. So through all these different models, so from the experimentation we do with Train the Trainer and the pilots we did with the distributed classroom models, we are now collaborating with other groups to further do research around these models, and or even general sort of like concepts as a way of increasing that capacity to deliver genomics training. So one of the projects that we are currently quite deep in, and we're about to run a course in October coming up, is one where we are going to be doing a train the trainer, particularly focused on pathogen genomics and for healthcare workforce in the UK. But we hope that we will run this a couple more times and we can open it up to other people globally. We've also been collaborating with Cambridge University and the COG UK on the COG-Train project, which is aimed at building capacity for SARS-CoV-2 genomics. And this includes using the online MOOC platform, the distributed classroom models, and of course, the virtual classroom models as well. And we're also planning in Train the Trainer, establishing capacity building that we're planning to collaborate with Africa CDC. We've also just completed a project where we were piloting a network, and this network was for AMR Genomics, which has been very successful in collaborating with the National Institute of Communicable Disease in South Africa. And the idea was to work with to try and establish training resources, as well as actually just establishing a network or a space or a platform where scientists can come together and start networking, which is what we hope should happen actually in a lot of the training programs that we have been developing. So these are some of the sort of offshoots of some of the ideas and platforms that we've been developing over time. So just want to touch on professional skills development, which is the third part of our strategies. So we have been, inundated with requests for professional. How can we get training on developing skills for grant writing, for presentation, for publications, and so forth. And in fact, when we did a needs survey for this project, that the AMR network, one of the top priorities for the network participants was that they want more training in grant writing. So we have actually provided a grant writing small workshops, and actually just looking at this publication where it's really clear that it's having scientists knowing all the skills, but without the professional skills is not really going to increase their capabilities or capacity. So we've also then gone ahead and developed a very nice resource, a podcast, which is focused on mentorship and career development. And this is Your Digital Mentor podcast, which is which is now reaching over, I think the number must be much more than this. This was a few months ago, over 115 countries. And Your Digital Mentor is really, we invite experts and we interview them around mentorship, around careers and professional development skills. And it's a unique podcast because we talk to people about their experiences around specific issues. It could be, skills, certain skills development from, how to write, how to present, entrepreneurship, and so forth. But these are real stories with people that are in the field and who have, seen it all, maybe seen a lot, and are willing to share their experiences. And so we've been quite successful, reaching quite a lot of people. And so if you haven't done so already, I encourage you to go and search on your podcast app, Your Digital Mentor podcast, and listen to our latest episodes. So season 3 will be coming soon. It will be coming in 2023. So you can, for those of you who've already listened to season 1 and 2, you can look forward to season 3 coming up. So if I've said all these things, I just wanted to say that it's not always so rosy. It's not, it's always, we're always going to be, looking at how do we address some of these challenges and issues. So we have all sorts of issues that we face, and this is just a I guess, a summary of some of the experiences we have had as a program that's developing training and capacity building, from issues around how do we get dedicated teams that are able to, that we can work with, because we all know everyone is busy. So expert trainers are not always easy to come by. Funding and resources is always an issue, because although we want to reach many people, we're always going to be quite limited as to what we can do, because of budgets, because of other issues, including pandemic, where we couldn't even travel, for example. We've also been battling with the idea of how can we continue to build those networks, because a lot of participants who come through our doors, they go back to their institutions and they're isolated. So one of the reasons why we started the AMR network was, how can we get people to start talking to each other? Because sometimes they're coming from the same country, same region, but they've actually never talked. They have the same problems. And so how do we facilitate networks? And also, how do we facilitate mentorship? How can we do that? And we also contributing to discussions around that, because I think everybody's battling with that idea, with that problem. So there are other barriers, cultural issues, such as, and also language. We also, want to know how can we make sure that we are able to empower other scientists or other upcoming scientists that are interested in developing this side of things, but also working with people in their institutions to be given the resources and support that they need in order to develop training and to have sustainable programs in their institutions. We also have been, we want all this training and all these programs to be representative. And so looking at EDI issues and how do we implement policies that ensure that we are being equitable, we are being inclusive, and so forth. So all these are some of the issues that I think have to be included as you're designing your capacity building program. So here's the part where I say, if you haven't heard anything I've said, or if you've forgotten everything I said, it's okay, because I am just now going to summarize all that into this slide. So here, what I would like to propose, and this is just a framework that I've come up with, and I think it could be relevant for African Biogenome. This is a framework for effective training and capacity building, and it can be applied to any capacity building program, I think, is that you obviously want to make sure that you develop the actual skills of the particular topic or subject that you're interested in. In this case, it's genomics. You want to think about the content, the delivery formats, and organization. You need to think about how are you going to continue to develop that capacity to deliver, and making sure that whatever you're putting in place is going to be sustainable. How are you going to be thinking about scaling up? How are you going to be increasing access, and so forth? It's important to have a good sense of of what's going on, how to collect information that helps you to increase the impact and understanding training needs for the target audience, and also stakeholders, including the funders and so forth. And so the research and evaluation, so just some of the examples that I've talked about earlier, but particularly and absolutely important are the professional skills development, because you need to empower people to be able to, to be, active out there through things like networks, for example, mentorship, but also all those other professional skills such as grant writing, publications, and so forth. So a program for capacity building cannot be complete if it doesn't have an arm of professional skills development. But we have to remember, obviously, that equity, diversity, and inclusion are going to be key across all these different activities. And what this means is different for different groups, for different teams, but you must lay that out right from the beginning and make sure that all the people understand what that is and also how to measure it. So having a policy that that everybody understands is really important. So, for example, we have a policy, and of course, it comes with these challenges on how to implement it. But you need to be acting positively in making sure that you are doing such programs with the right mindset, with the right culture. And also just to make sure that within the leadership and the people that make decisions are also a diverse group, and that people are consulted and included in making the decisions about such programs. And of course, finally, sustainability strategies are always, important to think about. So, how are you making sure that you always have enough trainers all the time? How are you thinking about who's going to be leading in those training programs and in those issues? But obviously, having collaborative partnerships can also extend the lifeline for such a program and fundraising. So that's my summary. And before I conclude, I would like to invite you to complete a survey, which we have out at the moment, Equity in Training survey. So if you could take out your cameras and scan the QR code, please go ahead, and we can share the link with you afterwards as well, if you are unable to pick up on the link now. And finally, I would like to acknowledge the learning and training team who contribute every day tremendously to the work that we do, from our director, Michelle Bishop, our admin teams, our IT teams, and others. And also finally, just to acknowledge some of our really strong partners and projects that we are currently quite busy working on. That includes the T3 Connect project, the Cogtrain, the H2O Bionet. We also work closely with EBI, as well as Wellcome Sang Institute. So that's it from me, and welcome any questions.
Thank you so very much, Alice, for giving us insights to this and talking on training and capacity building strategies for global genomic training. So Alice is from Wellcome Community Science, United Kingdom. Thank you so much for your presentation. So at this time, we'd like to welcome questions for Alice. Does anybody have a question to ask Alice? Indicate by raising your hand up. Okay, please proceed to.
Thank you, Alice, for the presentation. I would like to know I know you've talked more about the health side. Are there efforts to make inroads into plants and animals and other microbes as well, that are kind of non-human systems? That is one. Secondly, when you deliver trainings in Africa, how do you will I say communicate to the people, like in French-speaking countries and in Portuguese? I mean, how does that work in general?
Yeah, thank you for the question. So yes, actually, we have been, we have been venturing into sort of non-pathogen or with the common pathogen or non-human genomics. I have to admit that in terms of the courses side, it's been a bit slower, but I think some of you might already know about the project which we're working with, Sujay, and I think another part of the team working on the Tree of Life kind of training to use some of the tools that are important for genomic analysis. So indeed, it's an area that we have, we do work with scientists, and it's still a bit early days for us, but there have been quite a lot of collaboration with those communities within the conferences side, so that Stressor Craven has been working with some conservation groups and also others as well. So yes, indeed, we'd like to hear from you, and that's why we are here. That's why we are happy to contribute to this, because we hope to connect with such communities. And then the second thing, which was, how do we communicate with people? Oh yes, you about the language. Yes. So actually, we mostly all our training is in English, and we have. I mean, we do sometimes, have, not like French-speaking trainers on our teams and so forth. But we also realize that that's not enough. And so we've been, we're actually going to be thinking how we can improve, access through language. And it's actually one of the questions in the survey I've just shared is asking about that as well, about the languages that people are using in their training. So we also would like to hear from you how you think this can be improved, because we want to work with people from different regions and access more people, and particularly in French-speaking countries. And we do realize that sometimes we don't access those regions as well as we do in the English-speaking countries. And therefore, we, again, would like to work with more scientists and experts that are coming from those regions, so that we can collaborate to develop those resources that can be translated into different languages, for example, but also even be part of our training teams, so that we can include that.
Thank you so much, Alice, for addressing that question. So I'd like to know if people still have questions that they want to ask Alice. Please indicate, let me know.
Okay, well, yeah. Thanks, thanks everyone.
Okay, so yeah, thank you, thank you,
I
Alice.
will paste my email address in the chat if anyone wants to reach out to me directly. Thank you.
Yeah,
thanks,
thank
Tyler.
you. Thank you very much, Alice, for your presentation. Yeah. So next we are going to take the next presentation, which is supposed to come before now, but I'm sure that Charlotte is now on the call, and Charlotte is going to be giving us a talk on the African BioGenome project and the African Open Institute for Genomics and Bioinformatics. Charlotte, are you on the call? Please share your screen and give us a presentation. Hello, Charlotte. Okay. So Charlotte is joining the call, but then we can have a 2 minutes break, and then Charlotte will join the call, and then we'll continue.
So maybe while we're waiting for Charlotte, Alice, could you please quickly take the question in the chat box? This is for you. Yeah, type just now.
Yeah.
Okay. Yes, sure. Thanks, Crystal, for the question. So actually, you can access it on any podcast app. So it's not just SoundCloud. You can go on YouTube. Just search for Your Digital Mentor podcast or Google Podcasts, or any podcast app that you normally use. Apple Podcasts, for example, you should be able to access that. Yeah. Thank you.
Yeah. Thank you. So, Charlotte, are you on the call now?
So I think, can we move to the next presenter, please, Taro?
Yes, I think that's what we're going to do. So, thank you again. Charlotte is not yet on the call, so we'll proceed to take the next presentation. And this is going to be given by Kim Pruitt, and she is going to be talking on the overview of NCBI genome resources supporting biodiversity research. So at this time, I'd like to welcome Kim to give a presentation. Kim, are you on the call, please? The floor is now yours. Hello, Kim.
Hi, can you hear me?
Yes.
Okay, I am setting up my screen to share.
Yeah.
Let me see if this works. Do you see my slide?
Yes.
Okay. Okay, good. I want to start by saying thank you very much. I'm really pleased to be here and to learn more about your program and to share some resources that we have that possibly will be useful to some of you. So I'm going to do something I rarely do. I'm going to show you, I have a mixture of slides and live demo. So all of you keep your fingers crossed for me that my live demo works just fine. So I'm going to start with some background information. Hang on. There we go. I skipped a slide. Nope. Okay, so here's background information. So at NCBI, we're known for having a lot of data. And so I'm focusing on the sequence data here. And data can be categorized to us as sort of the primary data, the assembled sequences that are available in GenBank, or the raw sequence data that's emitted directly from sequencing platforms that's available in the Sequence Read archive. Can you see my mouse cursor moving as a pointer?
Yes.
Okay, good. We also have a project at NCBI called RefSeq for reference sequences. And RefSeq is something that we build based on the data that's in GenBank and SRA. And in the next slide, I'll give you a little bit more information about RefSeq. And then on the right, this is all of the metadata that we hold, ranging from information about the organism's scientific name, to descriptions of the biological samples, to descriptions of the genome assembly, such as quality statistics, the number of chromosomes, whether it's assembled to the level of a chromosome. And, oops, sorry. And also gene information, gene names, functional information when we have it. Unique persistent identifiers are provided for each of these data resources. And that's important to be able to cite the data resources and give credit to the people that have submitted to us. And so, we're strong advocates for people getting that acknowledgement and having provenance for who submitted the information to us. And then data and metadata are indexed to support search and to support cross-linking between these resources. Quickly, the major difference between GenBank genomes and RefSeq genomes is that GenBank data, it's submitted. I mean, this is the data that is being submitted to NCBI by international submitters. That is the result of primary research. RefSeq, as I said, this is a project that we have taken on ourselves, and it's built from the combination of analyzing GenBank and SRA data. It's supported by curation and QC. Another major difference between RefSeq and GenBank is the redundancy, the size of the holdings. GenBank, of course, has everything that's ever been submitted to NCBI or to our partners in the International Nucleotide Sequence Database Collaboration, INSDC. And so that includes, for example, partners in the UK at EMBL EBI, and in Japan, the DDBJ database. GenBank genomes come in sometimes with annotation and sometimes without. Whereas RefSeq is basically, it's an annotation resource, and 100% of the RefSeq genomes are annotated. And of course, there's just a big difference in terms of the size, the number of genomes that we have in GenBank. This is just a eukaryotic count, is over 25,000. For RefSeq, the eukaryotic count is over 1,400. So I want to introduce to you today a new initiative that we've recently embarked on called the Comparative Genomics Resource. The resources that I just mentioned to you, they're sort of siloed. You have to know to go to this database or that database in order to find information. And the Comparative Genomics Resource is pulling that together so that it's easier to navigate across the data that we hold. But we're also, as part of this initiative, building out some of our tools that were either initially provided for GenBank that were used in the submission workflow, or that were used in deriving RefSeq genome annotation. And so we're building out our tools to make them publicly available. So you don't have to have your data here in order to use these tools in the future. You'll be able to just install the tools locally, and some of that is what I'm going to be telling you about today. So the Comparative Genomics Resource, and I think here is where I try to transition to my live demo. So hang on a second. Can you see my web browser now?
Yes.
I hope. Okay, so you find the Comparative Genomics Resource by just remembering the acronym CGR. You plop it in, you do a Google search NCBI CGR, or you just search CGR on the NCBI webpage. And this brings you to the Comparative Genomics Resource landing page. And so I'm going to be focused on this page. There's a lot of information. You can subscribe to get updates about what's going on in this space. And so you get updates about what tools are being released. Documentation to explain the initiative, how to get involved. But I'm going to be focusing on these last three panels here and showing you some of our data resources, analysis tools, and data quality tools. So if you go in to this page, data resources, I'm focusing here on the very top thing, NCBI data sets. So I'm going to scroll to different tabs that I've pre-set up. So NCBI data sets is a place to download data. And so here I'm going to put in African grass rat and select. You can put in a common name or a scientific name. As you see, I've put in a common name. And here's information about the taxonomic, if you wanted to find the identifier for the taxonomy, you can get there that way. We also have here, I'm going to show you access to genome data. You can do batch uploads of a list of NCBI gene IDs to get ortholog or gene information. There's a command line interface that you can install locally to use, and there's extensive documentation that I invite you to explore. So if we go into the genome interface, and I'll again search for, I'll search for Cassava in this instance. So what you're seeing here is a table of the assembled genomes that we have at NCBI for Cassava. And so the top row is a case where we have both a RefSeq annotated genome, and in this case, the GenBank genome also carries annotation that was submitted to by the submitting organization. And so here's a case where there's annotation for both. For RefSeq, we typically pick one best exemplar based on the level of assembly. Here it's at a chromosome level and some of the quality metrics for that assembly. The rows below are showing assemblies that do not have any annotation, but the genome sequence is available for analysis. And you can select additional columns to show on this display by just simply clicking here. And you can select one or many genomes that you would like to download locally. And you can either download the table that has all of the columns that you've configured to include in it, or you can download the package. And when you download the package, you can decide that you only want GenBank, or you only want RefSeq, or you want both. And you can pick what you want to download, just the FASTA. Do you want the annotated features? And here it's in GFF or GTF format. Do you want just the protein FASTA? So you get to select what information that you want to download, and you can name the file down here so that it's something that is more meaningful to you than our default name. So I want to show you briefly let me change my settings to share sound, just a minute, share sound. So I want to show you a YouTube video. If the sound doesn't come through, please let me know. But this is a YouTube video illustrating accessing NCBI datasets through Galaxy. And so I've heard that there is a Galaxy Africa platform, and so you should be able to access this in the Galaxy
platform. This is a quick introduction
platform.
to using NCBI's datasets resource within the Galaxy project.
Let me know if you can't hear the
Whether at
audio.
NCBI or in Galaxy, datasets allows you to download sequences and metadata for genomes, transcripts, and proteins, either in a browser or from the command line. Datasets currently provides access to more than 24,000 eukaryotic genomes, 1.2 million bacteria, 10,000 archaea, and 50,000 viruses. Let's see how you can access all of these genomes within Galaxy. This is the public server page for Galaxy, usegalaxy.org, and it's advantageous to have an account so that you can save workflows and results and access them from anywhere. On the server page, you see 3 panels. On the left, there's a list of available tools. On the right is the history panel. In this case, we're starting with an empty history. And the center panel is where you interact with the tools and data. Let's find the NCBI Datasets tool. You could, of course, search for the name NCBI Datasets, or find it under Get Data. When you click on NCBI Datasets Genomes, the tool appears in the central panel. The default option here is to download genomes by accession, but you can choose the taxon option instead, which accepts virtually any taxon level, and we'll be downloading data for the Ugandan red colobus, Piliocolobus tephroceles, in just a bit. The Filters and Limits menu lets you choose the source of the assemblies, so you can limit to reference genomes, for example, or only genomes with annotation. And the File Choices menu allows you to customize the data package to get only the file types you want. I'll get the default package by clicking the Execute button. The central panel now shows that the job was submitted successfully, and on the right we see the files that are being downloaded. The gray color indicates that jobs are queued. Once they start running, the color changes to orange. And if the jobs are successful, the color changes to green. In Galaxy, files are organized by type. We have 2 metadata reports in JSON format, a data report which includes metadata for all genomes downloaded, and then 1 sequence data report per assembly. We also have the FASTA sequences and an annotation report in GFF3 format. We downloaded only 1 genome, but if we had requested multiple genome assemblies, each file would be identified by the assembly accession number. Let's take a closer look at the genomic CDS FASTA file. By the way, this is the assembly accession number for our genome today. Note that you can download these files for local workflows or perform your analyses within Galaxy. And there are several Galaxy tools that benefit from a reference genome retrieved with NCBI datasets. Under genomic analysis, you have annotation tools like Maker, mapping tools such as Minimap2 and BWA-MEM2, and RNA-seq tools like StringTie for transcript assembly. That's our quick introduction. Galaxy offers many help options, as does datasets, and you can provide feedback and ask us questions by using the feedback button or writing to info@ncbi.nlm.nih.gov. NCBI Datasets is part of the NIH Comparative Genomics Resource, or CGR, which is an NLM project to facilitate comparative genomics analyses for all eukaryotic organisms.
Okay, so that's data sets. So I want to move on to a resource called the Genome Data Viewer. And if I go back to our Comparative Genomes Resource landing page, you can access that through the Analysis Tools section. So let me find our way. So here, the landing page for the Genome Data Viewer provides an interoperable navigation tree where you can expand nodes to find the information that you're looking for, and then click on something. And on the right side, you can do a search or use one of the pre-canned examples in order to go to the browser. So I've pre-configured a view here, again, for the African grass rat. And on the top, so this is basically NCBI's genome browser. I mean, it's a genome browser just as UCSC has a genome browser or Ensemble has a genome browser, and this is NCBI's version of it. So on the top, you can see the species information, the name and accession number of the assembly. In this case, I'm looking at chromosome 6, and what I'm looking at here is a reference sequence record. And I know it's a RefSeq record because the accession number includes an underscore. GenBank accessions for individual chromosomes do not include an underscore, and RefSeq does for the individual chromosome accession number. On the top here, you can see the genes that NCBI has annotated, and this is all of the transcript variants. This view shows all the transcript variants collapsed into this version. For this genome, we also imported the Ensemble annotation, which you can see below for comparison. And we're showing RNA-seq coverage from our analysis. This RNA-seq coverage plus human or mouse transcripts may have gone into coming up with the annotation that's shown here. The bottom row is showing the intron features that we deduce from the aligned RNA-seq reads. And so the gaps here are the potential exon features. On the left, there's a navigation panel. If you know you want to jump over to a different chromosome, you just simply click on the icon. And on the lower part, you can add tracks. You can look at your history. You can add a BLAST illustration too. I'll show you that in another view. You can modify this view by clicking on tracks and then configure tracks. And in this case, I'm going to modify the NCBI gene view. And instead of merging all of the transcripts and coding regions into one collapsed thing, I'm going to merge transcript and CDS pairs and then update to configure that view. And you can see that we've predicted 3 different transcripts for the TP53 gene in the African grass rat genome. So I'm going to go over to a next tab to show you what it looks like if you add BLAST. So the way I provided this view was I went into NCBI's BLAST resource and I selected I searched for the African grass rat genome, and I entered in transcript accession numbers to align to the genome. So I put in 2 transcript accession numbers, and what you're seeing here is one of them in the first 2 panels and the second one in the next 2 panels. So the top panel is showing you all of the BLAST hits strung together into intron-exon type of view. And then in the next panel, it's each individual hit shown to you without it being interpreted into this assembled display. As you hover over these items in our genome browser, you get an info panel that will give you more information about the item that you're looking at. And that's true for all of the tracks. And as you can see, there's a lot of information here, really too much. I could spend a whole time talking just about this. And so this is all I'm going to show you because I want to show you one more thing. This central piece here is what we call the sequence viewer. And this central piece is available for export. So you could install it into your own web page if you wanted to. And you just look for NCBI sequence viewer or write to info@NCBI to find out information about how to get documentation for how to install it into one of your own displays. So I'm going to shift now. And again, going through this analysis tools panel, I'm going to shift now to something that's really brand new that we just did a beta release for called the Comparative Genome Viewer, or CGV. And the Comparative Genome Viewer, we're still working on populating the data. And so here I'm going to use mouse as an example. And so you can select a species in the first panel. And then you can select, in some cases, as I said, we're still populating data. You can select a second species where you want to see an alignment between the genomes, the whole genomes of these 2 species, or you can pick mouse, house mouse again, and you can pick different versions of an assembly over time to look at. And so you might want to compare the most recent version to an older version, and you set up your table here on this page, and then it results in a view that looks like this. And so this is Mus musculus, the most recent reference assembly, GRCM39, compared to Mus pahari. And you can see these lines are showing you where there's a region of Mus pahari on chromosome 1 that is aligned to Mus musculus on chromosome 19. You can click on these regions and you can zoom in. And so, for example, I've zoomed in to Mus pahari chromosome 11 in this view, and you can see that there's a small region that maps to Mus musculus chromosome 7, a large and 2 larger regions that map to Mus musculus chromosome 13 and 15. So now I'm going to transition back to my slides and wrap it up. And let me get past the things I put in here in case that didn't work. So I'd like to wrap it up by just telling you about something else that we've just released, a foreign contamination screening tool. And I want to give you some information about why we prioritized getting this out for people to use. So GenBank submissions that we receive, they come in. We currently do a contamination screening of all eukaryotic submissions. 64% of those received in our analysis come back with contamination, and then we work with the submitter. We let them know about the contamination, and in most cases they choose to, they agree with it and they choose to remove those contacts from their genome. 80% of them lack annotation, and of the 20% that do have annotation, over half are annotated with non-informative protein names, such as just hypothetical protein. So we were very concerned about the level of genomes that were coming in with contamination. And so what we did is we undertook to make our foreign contamination screening tool better and release it publicly so that people can use it before they reach the point of submitting to whatever repository you choose to submit to. And so our goals are to make it easy, reliable, fast, good, high sensitivity and specificity, and portable. And so this is sort of, what's in my tube. I sequence something, and what is it that's included in my sequence data? So we have two types of screenings. One is for adapter sequences, and one is for other organisms, ranging from bacteria to other types of animals or plants. These two paths are integrated with an ability then to clean out, it provides a report, then you can clean out the contaminating sequences if you agree with those reports, and then you can carry on your annotation and subsequent analysis. This has been publicly released, and so you can access it on our NCBI GitHub site for testing, and we're eager to find people to work with to further improve this tool. And on that, I hope I'm not too far over time. I want to thank you very much for your attention, and I would really love to take any questions that you may have. And thank you. And feel free to contact me afterwards too with questions if you prefer that over the NCBI help desk.
Yeah, thank you, Kim. That was a very nice one. So Taiwo? Yeah, I think we have some questions on the chat box. Someone said, can they tell about Mega? So I'm not quite sure if that is related. Is there something you want to expand about?
Can you elaborate? What do you mean by Mega?
Okay, so that is Mebin. So I'm just going to see if I can give Mebin. So Mebin, I can give you, maybe I can unmute you. So where are you? I'm just trying to find you on the call so you can elaborate more on this. So I'm just, there are plenty of names, so I'm trying to skim through. So do you mind typing your question, maybe? So just a moment. Okay, now I found maybe. Okay, I'm just going to unmute you. Do you want to quickly ask your question orally or verbally? Major Bean. Yeah.
Yes, sir. Hello, ma'am.
Hi.
Thank you so much for such a useful and sourceful information about NCBI. I'm a PhD scholar, and I'm currently doing my research. And my first objective is research is completely based on NCBI. Ma'am, after finding out the genes and sequences of Triticum aestivum, I am working on wheat plant. I have got all the sequence and details about that phylogenetic tree, but I want to make a analysis analysis, evolutionary analysis of that Triticum aestivum with Arabidopsis plant, Zea, maize, and Oryza sativa. For that, ma'am, I want to create a phylogenetic tree. So, ma'am, can you please tell me means how to create that with that software means like the Mega, so that I can make a evolutionary relationship among those plants and species?
If Mega is a software tool that I don't think NCBI maintains.
No, ma'am. Actually, it is not present in NCBI. I have found the details. Means I have found the gene ID. I have found the sequence ID. Everything I have found from the means the NCBI database. But after getting the sequence, I have to attach for creating the phylogenetic analysis. How that plant gene sequence is related to the
Yeah.
other plants.
So Major B, I think this might this is something that could be explored outside the workshop. Maybe you could potentially email Kim, and then I don't know if this is something Kim might want to discuss. Kim, what do you think? I mean, does NCBI maintain the Mega software? Because I used to think it's an independent software. Is it integrated or something?
It's not. So it's not something I'm familiar with, so I don't feel like I'm the best person to really address your question. I'm so sorry.
I would imagine. So Major Bin, what you might want to do is, if you try to reach out to the developers of the Mega software, then I think that would be very helpful. Yeah.
Okay,
So that's,
okay, thank you so much.
Okay. Any other question? Okay, people are asking if they can get your presentation, Kim. Are you happy to share this with the community?
I am. So I think what I'll do is I'll share it with the meeting organizers and then you can redistribute it.
Okay.
And so my slides, would that work for you?
Yeah, absolutely.
Okay.
That would work. So I've got some questions coming in from the live stream. So I also have one here. So Genevieve is asking, so I'm just gonna, maybe I can't find Genevieve on the chat, but she's asking, based on your experience, would you suggest we use the annotated reference genome uploaded by the submitters or authors, or the new NCBI generated annotated reference genome when wanting to do transcriptomic study? So Kim, do you want to take that?
So sure. So you could, that's a hard question to answer because it depends on the quality of each. And so with our RefSeq genome annotation process, we do use the same method across all of our genomes. And so RefSeq is inherently more comparable to other RefSeq because the same method was applied to annotate all of the RefSeq eukaryotic genomes. And we can't say, you'd have to look into the publication associated with the annotation that was provided by the submitting organization and determine how the quality of that annotation. And so you could, for example, use the comparative genome view that I showed you, where it's a beta release and we're actively working to add more data to that. You can request adding data to that to support your analysis. What I didn't show you is you can drill down to the level of the genes. And so it's possible to get some information comparatively using the comparative genome viewer, so you can see something about how the genes differ between the two. And with the NCBI data, you're able to use our genome data viewer to be able to assess RNA-seq reads that support the annotation that we've predicted. But with most whole genome annotations, it's been predicted based on alignment evidence of some sort or another. And so generally methods are pretty similar, but the differences in all of the heuristics that are being applied. So we like to think that the NCBI generated RefSeq annotation is generally of good quality. We have put in a lot of effort to try to improve on that annotation pipeline over decades now, and so it's a very mature process. And one of the things I forgot to mention is that we are planning to release our annotation pipeline publicly for public use. Right now, the only way to get your genome annotated in RefSeq is to contact us, or we, in cases, we may just have already selected it to put into our annotation pipeline. But in the coming couple of years, we hope that that whole pipeline will be available really for anybody to use. So we're excited about that. So if you want, you can contact us with a particular question where we might be able to give you more specific advice based on the taxonomy of the data that you're looking at.
Yeah, thank you, Kim. So Genevieve, please do contact Kim for further information. So we have some hands raised up. Adamu, I'm gonna ask you to unmute so you can ask your question. Adamu Idris.
Hi, I'm Adam Idris Lima from Nigeria. My question is, I'm trying to avail the genome data of SARS-CoV-2 from NCBI. So I want to construct phylogenetic tree. So the software, I need the metadata, but I couldn't avail the metadata. So I want you to help me tell me how can I avail the metadata whenever I'm trying to avail the genome data.
Okay, so you're trying to determine what data you need to download from NCBI? Is that?
Yes, it's genome of the SARS-CoV-2.
So I'm not familiar with Mega and what kind of what data imports it needs. So I think for your case, it may be best if you try to contact us with your specific example, and we can help you with that offline from this meeting. And you can contact me, and I can forward it to the right people, or you can contact the NCBI help desk at info@ncbi. So I don't think I have enough information to help you right here in real time. Sorry about that.
Okay.
Yeah.
All right. Thank you, Adam.
Thank you.
Okay, we have another Sujay Kumar from the Sangha Institute.
Hi, Kim. Thank you very much. Can I just check quickly how long, because I'm interested in contaminants and contaminant removal, and that's what I'm talking about tomorrow. How long, because I really want to use FCS or similar tools. How long, do you know how long it takes on average for a 1 gigabit?
Oh, you know what? I don't have those numbers in my head. It's pretty fast.
Because it's using Kamer seed matches.
It's Kamer based. Yes, it is definitely Kamer based.
And is it checking all genomes, or is it checking some genomes when it's looking for contaminants?
No, so we're looking very broadly and deeply. So we're looking for prokaryotics, viruses, other cross-contaminating eukaryotes. So we're looking very broadly. We have a very large Kamer library that we're checking against.
Cool. Okay. Thank you.
Yeah. So yeah, we'd love to have you give it a try and give us some feedback, because we're really trying to make it a very portable, robust tool. And so we're really interested in feedback.
Thank you.
Yep.
All right. Thank you, Kim. I think, and thank you, Sujay. So Kim, please do share your slides so it can be shared with
I
others.
will. Yep.
Okay. We really need to move to the next item. So we have Charlotte back. So Charlotte, do you want to quickly give a rundown so that we can move to Andre Egan?
Hi, everyone. I'm very sorry for the whole technical issue that I had down here. So I apologize for that, and I will just be sharing my screen in a minute. It's open in the background, and let me just get help with that, please.
So maybe Charlotte, shall we move on and then we come back to you later, please? If that's okay. Charlotte?
Yes.
Shall we move on and come back to you later?
No, I'm just opening my slide.
Okay.
Now.
I think you need to unmute.
Okay, so thank you.
Can you put it in a slide show mode? So
Yeah.
go to slideshow. Yeah.
I have this. I clicked on slideshow. Okay. So thank you so much for having me here at this time when I should have been opening the. I have to apologize. I had a meeting earlier than this one, and actually thought I was going to be presenting by 3. So I apologize for that. Still, I hope you enjoy my talk. It's going to be very brief. So I'm just going to be talking about the Africa. I hope you can all see and hear me. Please.
Yes, go for it.
I'm going to be talking about the Africa. Great, because yes, I'm going to be talking about the Africa BioGenome project and the AfricaBP Open Institute for Genomics and Bioinformatics. The next couple of slides. So, sorry about that. So specifically, the origin and timeline of the AfricaBP dates back to October 2020. At that time, the first stakeholder workshops meeting held at the Biodiversity Genomics conference at the Sanger Institute in the UK. So exactly 9 months later, in June 2021, the AfricaBP pilot was birthed, and this grew mostly through electronic dissemination. So in March 2022, our first position paper was published in Nature. And just last month, in August, AfricaBP recorded another milestone as it was institutionalized in South Africa in partnership with the SFA. So, sorry, I don't know why that keeps happening when I want to move to the next slide. What is the African BioGenome Project? In a nutshell, it is a coordinated Pan-African effort to build capacity and infrastructure to generate and deploy genomics data for the improvement and sustainable use of biodiversity and agriculture across Africa. This is under the community of networks called DAISY. That is the Digital Innovation in Africa for Sustainable Agri-Environment and Conservation. Now, the mission of this organization is to foster scientific collaborations and partnerships, provide a platform for innovations and policy change across Africa through biodiversity genomics. Our vision is to harness the power of genomics for the improvement and sustainable use of biodiversity across Africa. So, like I mentioned earlier, we already published our first position paper in Nature, and this was just March, earlier this year. So basically, we want to sequence 45,000 plants and about 60,000 animal genomes with a target of a minimum of 2,500 genomes per year. And we're doing this in collaboration with the 10KP and the VGP consortia. So since the publication of this position paper, the AfricaBP has received high-level invitations from, top-rank bodies like the United Nations, African Union, and recently the FAO. So the AfricaBP runs under 8 functional subcommittees from the sampling, collection and processing, sequencing and assembly, annotation down to the partnerships and fundraising committee. So we are growing in popularity. Yes, we are trending. And basically, we are growing our following via our social media platforms. But it is importantly to keep the public abreast of our programs. So feel free to visit our website to have a look at the project in details. We're on Twitter, LinkedIn, YouTube, Telegram, and still expanding. With the publication of our first position paper, we had over 5,000 visitors from 120 countries in the 6 continents. Please feel free to join any of our social platforms if you're not doing so already. So I'm just going to talk about the AfricaBP Open Institute. And again, I welcome you here again if you didn't join in our previous workshop, and this is your first. So
Yeah, I think we lost Charlotte. So just maybe give her a few seconds to see if the network comes back. Yeah, I think I can see she's frozen. Now we can't see her any longer again. Okay, Taiwo. Charlotte, are you still there? I think we lost Charlotte, so maybe we can prepare for the next presenter. That would be, I'm just gonna check the list. Andre Egan. So, Andre Egan, are you on the call today, please? I did say you actually, okay. Please go for it, okay. And then after your presentation and the questions, then we can take a break.
Can you hear me?
Yes, we can all hear you.
Okay, and I think the second question is, can you see my screen or my slides?
It's still coming up. Yeah, okay, it's good. It's like we can all see it now.
Okay. Thank you so much, and I'm happy to present this afternoon and follow actually the presentation by Kim and Charlotte, and be here as Illumina representative. I'm Andre Egan, working in agrigenomics for Europe, Middle East, and Africa. And prior to Illumina, I spent around 20 years of my time doing research in livestock genomics and also be part of the first sequencing project to sequence the bovine genome. Here, I'd like just to share some high-level thought and slide about what Illumina genomic technology and solution could be for the AfricaBP project. And just as a refresher about Illumina, we are a global company providing sequencing and array-based technology around the globe and serving different markets. So a large portion of Illumina is dedicated to human health and unlocking the human genome to improve human health. But as you can see here, we are also very much concerned, and that's where I'm fitting fully here with agriculture and what's happening in agriculture. And mostly because we know that one of the challenges is to feed the world in the future. And genomics, also, if it's not a magical tool, applying genomic approaches to existing resources will expand the ability to meet really some challenges that we have associated with food security. So that's clearly where we fully fit in this trend of feeding the world, with, I think, two major technological revolutions that happened over the past decades. One is on the cost of sequencing that is decreasing and will continue to decrease in the future. And that is really thanks to all the effort and all the improvement that were done as part of sequencing the first human genome and some project following that. And the second technological revolution that we were also part of is the ability to actually genotype, so place on a microarray some genetic variant that could be genotyped in parallel, not only a few at the same time, but thousands, 10,000s, or 100,000s at the same time. So these two technological revolutions really changed the way people are approaching and scientists are approaching this question of biodiversity and feeding the world. Of course, at Illumina, we are not just happy with those two key technological revolutions, but what we are also trying to do and to work on is to come up with some integrated sample to answer workflows. And so in order to leverage our technology and to have an easy way from when you get the biological samples till you are getting the first analysis done from the sequence. And so we are continuing to evolve our technology in different kind of applications. So I don't go into detail of what is needed to sequence or to produce a DNA sequence, but of course you need DNA. So that means that you need to extract DNA. And there is a step that is called library prep. And then you will use a sequencer to produce a sequence. And through this process, you need to be able to call the bases and then also to do some first analysis on maybe comparison with some existing sequence and building actually also some genome and genome assembly from scratch. So different kind of analysis that are performed. So what we are looking is really trying to facilitate and simplifying this entire workflow process from samples to answer. When we look, of course, on the use of genomics to secure the supply of food moving forward, a big part of what is needed is to better learn what is around. And that's where I think the African Biogenome project is also very appropriate, because before you can start using some of the existing species that we don't use today to feed the world, we need to learn more about their genome, learn more about how the genome is functioning, and what they could provide in terms of resource for food in the near future. And so that's where our technology and our sequencing technology especially is fitting in, as clearly when you can sequence in parallel a lot of, or if you can produce a lot of sequence in parallel, so with a throughput that was not even thinkable 20 years ago, that's where you can start making progress in knowing the genome first, so knowing and having a kind of rough reference genome or draft reference genome, but then you may not be happy with just knowing the genome and the DNA sequence in a genome, but what you want also to understand is how the genome is functioning, how the genome is actually maintaining an organism, how the genome is actually contributing to the growth of an organism, and also to some production traits that the organism is really very useful for. So knowing the genome function, knowing gene expression, gene regulation, all these different methodologies are built into our samples to answer workflow with different kind of library preps, with different kind of methodologies that people have created, have built on top of our sequencing methodology. So also, without taking much time on that, the best would be to just have a look on our website with all the different sequencing technology or methodologies that are existing and different workflow, from just producing a genome sequence to start building a reference genome to addressing the question of gene regulation or even differences in methylation. We have everything in place to do so. Once you have the genome, of course, you have a first genome, but you may be interested, and that's where, for example, a part of the biodiversity is interested in, is not just knowing the number of different species that are around, but what is exactly the genetic diversity, what is the biodiversity within a species. And for that, you would sequence and resequence different samples from the same species. And you would discover some new variants, and that's where clearly a sequencer like a NovaSeq would fit in to discover novel variants. And that's part of what we call the Illumina genomic continuum, because we have everything to discover variants, so produce sequence, discover variants, but then also make use of the variants that are found by building some SNP genotyping arrays that could be used then later on for GWAS study, or even in some species where genomics is very straightforward, like cattle, you can even do genomic selection at a very high throughput level. So a combination of discovery by producing more sequence and actually taking some informed decision based on the genotyping of different variants that were discovered on the genome. And just to give you an example on this Illumina genomic continuum, it's currently happening with a project that we were sponsoring through the Illumina Greater Good initiative. The winner was the International Camel Consortium with Elena Chiani from University of Bari. And the project is about sequencing 400 camelids, so an equivalent of 20 terabases, and analyzing the data, selecting, of course, discovering a lot of variants, millions of SNPs actually, and then selecting a good set of SNP of single nucleotide polymorphism on the genome to produce a first camelid bead chip. And so we are exactly in the process of discovering, then detecting the genetic or characterizing the genetic diversity and producing the tools so that camel breeders and people interested in camelids could actually have a tools to make some informed decision moving forward. We are, I mentioned, we are a global company, and we are quite well implemented with many different systems installed around the world. Of course, for the African Biogenome project, from what I understand and what I read, and what I discussed with different people, is that it is a project for Africa in Africa. And so here again, it's very critical to build also capacity in Africa. Before moving to what we are doing in terms of building capacity in Africa for sequencing and genotyping, let me just mention what we are doing in terms of facilitating or simplifying the entire workflow, and especially the analysis bioinformatic part of the work. I mentioned already before, when you extract the DNA, you need to sequence it, and then you have some analysis that needs to go on. And here we came up with a tool that is called Dragon for dynamic read analysis for genomics. That is really a collection of bioinformatic pipeline used for secondary analysis. So this is happening right after the sequencer, and it takes reads coming out of the sequencer, and it creates, in some cases, variants, if you look for differences in sequences, or it creates counts. So it all depends on the application. And there are quite some benefits of using these tools. It's very accurate, and we demonstrated that by different work we did with different institutes. It's comprehensive, and it's also very efficient. Just to give you also a flavor of what Dragon is. Dragon could be on the cloud, but also we thought very key is to have it also on instruments. So like on the NextSeq 1000 or 2000, Dragon is directly built in on the instrument, so that you don't have to share data over the cloud, which could be a problem if you don't have a decent internet connection or even a high-speed internet connection. So the right tools to do the secondary analysis part of analyzing sequence data. And we are coming always with updated version, but just to show you how fast it could be, the latest version of Dragon, so Dragon v4, can just call from a human whole genome. So that means here it's written 34x coverage. So from this human genome data, calling all the SNPs, so all the variants actually, and so making all this analysis could be done in 30 minutes per sample. So just demonstrating the efficiency of such a tool. We are also building in some machine learning, and this slide is way too busy, but just to highlight on the right side that there is a difference in running time between some using machine learning in Dragon v4, the light orange bar compared to the dark orange bar that was not using machine learning. And on the left, it's giving you some insight into the accuracy of using Dragon. And you see the latest version of Dragon will be the very first bar column on the left. And we see here how accurate this is compared to other technology or even other version of Dragon before. So just a tool that we will continue to expand and to promote so that people can have an easier way to access sequence and really to spend time more of the biological relevant part of the analysis and not wasting time with the first part of the secondary analysis. I mentioned that we are also involved in building capacity for Africa. And here I want to highlight, for example, our connection and strong connection to A3 Africa on the human side. And we actually work that an iScan, so a genotyping platform, could be placed in Africa for Africa and dedicated to human genetics. And this is currently running at CPGR in Cape Town. And we are expanding also our capacity, and this graph is showing how this expansion is going. If you compare what was available in Africa in terms of Illumina instrumentation in 2018 and what is available in 2022, that's clear that we had quite an expansion, and we will continue to move forward with that. You may have seen, but here an highlight on that we are also partnering and helping in some projects directly in Africa. And this is, for example, this pathogen genomic initiative that we committed with quite some an amount of money in sequencing capabilities to a global pathogen genomics initiative. And so here, this it should help clearly to build capacity labs in Africa, to build NGS platform, to have access to reagents, and also to get the appropriate training to perform the work for really a pathogen genomic network that is currently ongoing, or that is currently being built around the world, because that's critical also to public health. I mentioned before with the Camelid project that we have an Illumina Greater Good initiative running. And here to mention that people, if you are interested to submit a grant, please look at our website, and before the end of September, you can still apply to the Illumina Greater Good initiative. Some of the past recipients of the grant were, for example, Rajana from Nigeria, working on yams, and we are currently sequencing with Rajana around 600 different species of yams. Some other people that were past winner, for example, Apollinaire, about some viruses infecting cassava. We had also a project on the baobab tree, and that was reflected by my first slide of my talk. So all kind of different projects as part of the Illumina Greater Good initiative. So if you are interested, just go on to our website, look for that, and submit a project. There is the grand prize winner will receive up to $350,000 in-kind consumable or service at list price from us. So that's really a dedicated effort that we have to support research, enabling a more sustainable, nutritious food supply around the world, and especially in developing countries. I'd like to finish by mentioning two different projects that we are also involved that are that have something to do, I think, with the African Biogenome project, or that have are similar needs. One is an Oceanomics project. Oceanomics is actually a project that aims to build a genome reference library of 20,000 fish genomes. And so it's done by the Minderoo Foundation, and Illumina, and the press release came out earlier in August. The Minderoo Foundation, they have a ship, a boat, where they build a lab to travel on the ocean and collect samples, biological samples. It's a kind of eDNA approach where they are filtering what they are getting in terms of samples in the ocean. And then we work with the Minderoo Foundation to install on their boat an XSeq 2000 for them to perform the sequencing work, the first sequencing work on the boat. Why that? It's because it's cutting time of shipping all the samples and organizing the sample, the shipment of the samples on the Earth again. And so everything is sequenced first on the boat, and then they can kind of know what's needed to be performed in the future, and know also if the sample collection was efficient enough and have a first, really a first idea of what they were sampling, and do some first quantification also before actually sending the relevant other samples to their other lab to be sequenced on a NovaSeq 6000. So just an approach that is trying to stay as close connected to the field as possible while building some high throughput sequencing capacity. Another project that we are involved is about iConserve, or conservation genomics. And here I want to highlight the work by Magdalena and Thomas, Magdalena from Congo and Thomas from Spain, where they are building actually a database with genomics data from gorillas across different habitats, and in West Central Africa, but also North and South. And actually what they want is to be able to find again the original location in terms of, for example, trafficking. Where is the original location of that gorilla that was found, and where should it be placed back, so to go back to his original location, geographical location. So Magdalena and Thomas, and we are supporting this initiative also. They have also a goal to train local scientists, professors, students from the Marian Nugbi University, but also some other people around Africa, if needed, to use next generation sequencing and derived methodology to sequence future confiscated gorillas, and so and enable then authorities to act quickly in those cases. So really build an genomic ecosystem that could be aligned with the conservation of the genomic conservation of gorilla moving forward. So I think I don't want to take more of your time. I stop here, and I'm happy to take any questions, or you are also welcome to contact me directly. Here is my email, and I will come back to you as soon as I can. Thank you for your attention.
All right, thank you, Andre. Again, that was a good presentation. So I'm just going to see if anyone has question. Does anyone have question? Use the just raise up your hand using the icon, or comment in the chat box. Okay, we have the first question from Tim Newman. Go for it, please, Tim.
Yes, hi. Andre, thanks for that presentation. I wanted to ask you something that's been an issue. By the way, I represent Diplomics, which is a omics research infrastructure program in South Africa, and I'm based in Cape Town. Mm-hmm. My question for you is about enabling genomics, and especially next generation sequencing in Africa and South Africa in particular. And you showed a slide that indicated from 2018 to 2022, there were addition of 2 NovaSeqs. We actually have 3 NovaSeqs now in the country, and we have an additional 60 plus various NGS platforms from Illumina. But the ability to do next generation sequencing is actually not limited by sequencing instruments. It's limited by the high cost of the reagents and the consumables. And you mentioned a couple of programs, especially the Pathogen Genomic Initiative, where the package includes either a donation or a significant discount on the reagents. I have tried in the past with various other Illumina representatives to try to negotiate discounts on reagents and flow cells in particular, because we pay at least 30, 40, perhaps 50% more than anywhere else in the world, especially Europe and the US. And I've not met with any success. So I wondered if you have some suggestions on how we could work with Illumina to be able to lower that price barrier and the price penalty so that we can actually enable sequencing in the country. Mm-hmm. Thank you.
Yeah, thank you. Yeah, it's so in a country where we are not acting directly, there is always some additional costs that could come from a distributor or channel partners that we are working with, right? And so what we are trying to build, and if you look around the world, while in many different countries we are still indirect, we are, as soon as the level of business is high enough, we are also building all the direct capacity in the country. So I think that's also one way we have, if you look over the past few months, what we have done in South Africa, we hired people from Illumina that are belonging to Illumina. And so we're building this Illumina team in the country also as we move forward. So that should help, or that should be one factor to help. The other one is, I don't know the exact detail on the pathogen genomics initiative and where the reagents will go and under which condition, but I'm happy to work with you and see what is done exactly and how to access to that. So that would be fine. And then sequencing, I think there should be a few announcements coming on before the end of this year. We have the functional genomics Congress coming up next week or in a few days. And I think you heard our CEO speaking about decreasing the cost of sequencing moving forward. So we will continue to do that. But I'm cautious that, yes, at the moment there is a difference, and that's exactly what we discussed when I was in South Africa 2 weeks ago, the difference in pricing in reagents, and that if we want the work to be done in Africa, we have to find a way to really have also a pricing that is affordable for Africa. Otherwise, they will be continuing to send the samples out, which is really not what the goal and the objective of the African Biogenome project. So while I don't have all the answer to your question, I'm very, I know the problem, and I'm very committed also to see what we can do. And we have now a growing team of people that could help and that should help. And team, I'd like to investigate further. So we will be in touch. I will make you in touch with some of our key people, so that we can work that out.
Thank you very much.
Thank you, Tim.
I hope to have a conversation with you again.
Yeah, sure. Thank you.
Oh, thank you, Andre. So I have another question for you from, just a moment, there seems to be lots of questions here. Okay, this one is Kim talking to Sujai. Then we have a question from Yasmina. What is the size of the SV that can be detected with the Dragon 4 system?
So the size on the variant that can be detected by Dragon, actually, I would say the size is not limited by Dragon, but it will be limited by the data. So we can identify single nucleotide polymorphism, or you can detect also some structural variants if you have the appropriate sequence to do so.
Okay, thank you, Andre. So Yasmina, do you wanna talk more about this? Let me just see if I can give you the. So I'm gonna ask you to unmute Yasmina so you can comment.
Thank you, thank God. Thank you, Andre, for this very nice presentation. No, I was just wondering, I mean, given the limitations in the size of the reads from Illumina, so I guess it wouldn't compare with other platforms that do long reads. So I'm just wondering, what is the maximum size of structural variants that would be detected by your whatever sequencer available?
So it's still, I would say it depends on the sequence or on the data that you are getting, because you can, from short read, you can still align some large contig. And even we worked last year with UST on the TELSEEK library prep, so using linked read to get longer contig. And we announced also Infinity earlier this year, the Infinity methodology that would help and that would produce up to 10 KB sequences or up to 10 KB long sequences. So all that will be managed, of course, also by Dragon. So that's how we are pushing also the boundaries and bringing also innovation. So while, yes, I'm aware of the current, some current limitation with the long reads, but we are working on it. We work, as I said, with TELSEEK, and we will continue with Infinity. And I show some accuracy data using Dragon and compare with some other technology. So I think we are still confident that we are moving in the right direction here.
All right, thank you, Andre. Any more questions? Okay. So let me see if there's any more question for you. Thank you, team, for the comments. I think this is from Crystal. I am also based in Cape Town. So maybe Crystal, shall I ask you to comment on this, Crystal? So you'll be, can you please unmute? Crystal Tully, yeah.
Sorry, I don't know what you're asking. I haven't.
Yeah, you have a comment in the chat box. So
yeah, that was just a comment for Tim and for Andre to thank, just to sort of back up the discussion there about the costs and finding the same problems.
Mm-hmm. Okay, I think that's all with the comments. Thank you, Andre, and it was really informative.
Yeah, so, thanks to all of you. Thanks.
Yeah, we're gonna take a 10-minute break, and then we'll be back to listen to the next presenter. So that will be in the next 10 minutes. That should be 4:20 London time. Okay, hello. So, Aaron. Hello.
Hello.
Yeah. Okay, Aaron. The next.
Can you hear me?
Aaron, go for it. Yeah.
Can you hear me clearly and can you see my screen?
Yes, go ahead.
Okay. My name is Aaron. I work at AfricaBP as a technical support manager. Today I'm going to talk about how AfricaBP has been supporting genomic biodiversity studies research in Africa since 20 years. To give you a brief introduction about AfricaBP, the idea of the conception of AfricaBP came in 2000, but the company was started exactly 20 years ago, August 2002, by opening an oligo production facility and Sanger sequencing. And the company started to expand. In 2007, we were the first institute to give next generation sequencing services using Roche 545 platform. Then in 2010, we opened our first subsidiary in East Africa, which is situated in Nairobi, Kenya. Then we expanded more. In 2015, our first West African subsidiary was opened in Ibadan, Nigeria. Actually, that subsidiary has its own lab now. We started actually increasing our footprint in Africa. Our Ghana office was opened in 2018. In 2019, we opened our Tanzanian office. 2020, we partnered with PacBio to distribute the instruments and also acquired instruments, the PacBio instrument here, to give a hi-fi long-read sequencing, which I'm going to talk about it later. In 2022, we opened our offices in Cameroon, Uganda, and also Senegal. So you can see that AfricaBP have a nice footprint all over sub-Saharan African countries. This is our main office. We are based in Pretoria. As you can see, we have a big facility here. It also hosts a very good laboratory facilities, which I'm going to discuss about regarding the services that we offer at AfricaBP. This is our unique stuff. The company in 2002 started with only 4 people. Now we have about close to 150 members throughout African countries. You can see here our offices in different sub-Saharan African countries. We also are distributors of different brands, but I've mentioned here selected brands that we actually supply. One of them, the main one, is PacBio for long-read hi-fi sequencing. We also are distributors of Zymo Research. It's very important. As you can see, DNA extraction is the start of every kind of genetic study. Zymo Research supplies different extraction kits for normal DNA extraction and high molecular weight DNA extraction kits. And also we distribute reagents from New England Biolabs The same, again, you can get DNA extraction kits from NAB, also library prep kits for next generation sequencing. Circulomics is also for high molecular weight DNA extraction that's very important for long-read sequencing. We also offer software for bioinformatic analysis called DNA Star, and I'm sure you are all aware of Epinor for consumables. And also we have a company called that we distribute for called Trade for plastics and consumables. So what kind of services do we give at Inkaba Biotech? What can we offer in terms of genomic biodiversity studies? I would say our core service is oligosynthesis, because the company started by making oligonucleotides. We've grown from 2002 now to have a Mermaid 192 system, which can make about 192 oligonucleotides per day. These primers are synthesized with speed, accuracy, and purity. Primers are backbone of any genetic biodiversity studies. They're important for amplification, or they can be used as probes to bind for targeted sequencing. So it was important for us to supply these primers with speed, as there was an issue in early 2000 getting primers on time to do genetic studies. So this actually created a good solution for us to study genomic biodiversity in Africa. Last year, we also purchased another oligosynthesis machine called Mermaid 12 that we're going to be using this for making probes for genetic studies as well. Our next service is Sanger sequencing. We've got a DNA sequencing utilizing 2 API 3500 genetic analyzer. We've been giving Sanger sequencing services for the last 20 years. This is targeted DNA sequence analysis. We also give bacterial, fungal, and animal species identification. Part of the Sanger sequencing department is also a very specialized animal genetics department, which is called Zoomics. Zoomics works on any kind of domestic livestock or wild species animals in terms of actually health, different kind of mutation or DNA profiling or parentage analysis. So we have been offering this services since 2009. It just recently had a new name called Zoomics, which actually supports biodiversity studies as well. Part of the Sanger team is also have a fragment analysis team, which can be used for microsatellite analysis. This is very helpful for DNA fingerprinting. It could be used for healthcare, agricultural, and animal and conservative studies as well. The next service that we give is single nucleotide polymorphism analysis, SNP genotyping. This uses a very specialized a mass-based system that is very simple yet reproducible method. It uses a simple PCR-based primer extension method and a MALDI-TOF analysis, which can be used to analyze specific SNPs. For example, you've completed a full genome sequencing or genotyping by sequencing analysis. You've got selected SNP that you want to analyze for diversity analysis. You can select those SNPs and analyze them with this medium throughput instrument. So we have been giving the SNP genotyping services since 2015. And also the system can be used for epigenetics analysis as well. If you are interested in methylation analysis, that can be done with the system as well. Our key service that has been busy for the last 10 years or so is the next generation sequencing service. This is rigorously quality controlled, a high throughput sequencing workflow. So we have two different platforms, the Illumina short read chemistry platform. We use the Next 6500 for that. With that platform, we do a whole genome sequencing, a metagenomics analysis, an exome sequencing as well. But we've partnered with PacBio to have a sequel 2E system for long hi-fi read sequencing as well. So using the PacBio long hi-fi reads, we've been partnering with AfricaBP so far to sequence about 8 genomes, and we're busy collaborating as well, doing other full genome sequences using the system. So as INCABA has turned just 20 now, we've decided to partner with African BioGenome project, Genome Challenge for 2022. This is actually to officially, this will be officially announced tomorrow, to sponsor, to sequence data of 20 PacBio smart cells. That is close to 10 eukaryotic full genome sequencing. This will be advertised tomorrow, as I mentioned. So we have 4 broad criteria to select which genome that we're going to amplify with scientific relevance, the economic value of the project, conservational security, and cultural significance. So if you're actually interested in applying for this genome challenge, you can visit our website, or you can go to info@incababiotech.co.za. You can email and ask how you can enter in this exciting African BioGenome project and INCABA Biotech Genomic Challenge. So I have summarized what we do here at INCABA Biotech. I'm happy to answer any questions that you have. Thank you.
Thank you, Aaron. Does anyone have any question for Aaron, please? You can raise up your hand or type in the chat box. Any question for Aaron? So, Aaron, I was just gonna say if someone, just a minute. Okay. I'm just trying to check the messages.
While you're actually checking for questions, I just wanted to add something. In a way, the importance of having this kind of platform in Africa is very important. As I mentioned, we have a PacBio long-read sequencing system here at INCABA Biotech in Pretoria, and we also is in the process to have 2 systems in West Africa as well. As you know, as all of you know, for example, the Human Genome Project was completed about in 2003, but since then as well, most of the work is done on, let's say, an Eurocentric approach. So, and our population here in Africa is biodiverse, like we have a very diverse genomic here. So it's very important that we have our own genome database, and this genetic sequencing should also be done in African continent as well. So we very happy to support such systems, such projects. And it goes the same for plants and animal species as well. So that's what we stand for at Inkaba Biotech, to do the work in Africa with African expertise.
Yeah, thank you, Aaron. So I can't see any question, but one quick question is, I understand that Omni-C, High-C can be done with multiple platforms. I don't know. I mean, I'm not so regular in the laboratory side of things, but is that something that is that the PacBio platform does, and Nanopore and the NovoSeq? I mean, how does that work basically?
Okay, we are in the pipeline. I think Acclaim, my colleague, is also in the talk, but you might add something here with the Omni-C for High-C sequencing. It would require an Illumina platform, I believe, but we are in the process of actually doing some of the optimization in the lab. Acclaim, if you are in, maybe, I don't know if you can unmute Acclaim if he's in the chat, if he can add any point on High-C sequencing and what our plan at Inkaba Biotech.
Yes, yes. Can you hear me here?
Yeah, go ahead, please.
All right, good. Yeah, as we've mentioned before, so we are in the process of establishing the High-C workflow. So usually what it's required is before you do a high throughput, you need to run kind of a shallow run on either MiSeq or NextSeq. So in that place, just to determine if the sample really is good enough to go into a High-C library data production. So our goal basically is to, for one, do that shallow High-C run or QC step. And once we are determined or sure that the sample is good enough to produce good High-C library, then we up the scale to the NovaSeq. So we do have access to the NovaSeq in South Africa. So that's what we're going to utilize to generate data. So we can only do that on so the current workflows basically covers Illumina systems. So we're going to be using Illumina systems. So initially it's a 2C just to run it on a smaller platform. Mm-hmm. And once the data we show it's good, and then we up the scale to NovaSeq.
Okay, so yeah, I think because one thing is that there will be need to do the High-C, because that is like some of the things that the community is interested in as well, and in terms of scaffolding and things here. But I think as we begin to advance the genome side, which we can start being more clearer on some of these things really.
No, exactly. Yeah, so we have all the stuff set up. It's just a matter of getting green light from the AfricaBP community, or you or Prof. Olivia. So we've got everything set up just to do those QC runs on the samples that we have already generated the Hi-Fi data on. I think we have about 4 or 5, or including VGP species, like probably 8 species or 7 species that we've already sequenced. But the important thing here is when generating the High-C or Omni-C data, it's one has to take good care of the sample type that is being submitted. We might get away with it on generating the long read data, but coming to the High-C, it's a different ball game basically in terms of the type of tissue or sample you have. So from our side, we have all the stuff. We received all the stuff we needed to receive. It's just a matter of getting a green light.
Okay, thank you, Acclaim. So any other comments?
I think there's a question, Tankard, on the Inkaba Biotech African BioGenome project genome challenge. It's going to be officially announced tomorrow, and the submission deadline is 30th of November. There's still time to submit videos. So we are sponsoring 20 PacBio smart cells. That's an equivalent of sequencing 10 eukaryotic genomes. So it has to be non-human genome, and the criteria will be also announced. Then you just have to make a short video and submit that for the competition.
All right, thank you, Acclaim. Any more questions before I hand over to Taiwo? I think he's back, so that he can continue. I think we've finished the presentations. Taiwo, you want to take over? I think we're done, basically.
Yeah, thank you. Thank God for the effort, and thank you, Ira and Acclaim for the insight. But I'd like to just seek a clarification still on the CABA African Biogenome Project challenge, which I'd like you to explain. And this is when the genome has been sequenced. So do individuals have access to this genome to use them for something they would like to use? If, for example, someone who put in for the challenge got picked, whatever samples or species he or she is interested in, and then the genome for that species is then sequenced, do that individual have access to use the sequence for that species? That's something I'd like you to just give a little bit of insight.
So I think this will follow the typical procedure of sample collection and getting all the regulatory permits. And I think this would be enshrined in the, for instance, the material transfer agreement and all of the compliance with Nagoya protocol requirements and mutually agreed terms. I think all of those things will be decided in those documents. And of course, the idea is that the people or the custodian of the sample would be the owner of the sample and would ensure that they are the ones that submit it and they are the ones that own it. And of course, they would again have access to it, basically.
Thank you so much. And I want to thank everyone for participating in this workshop, the one. And don't forget that tomorrow is another day and we start at the same time. But at the same time, I hope that you have benefited from the presentations and you learned something today. So this is the effort by the African Biogenome Project to build capacity. So we are hoping that you can use this opportunity to gather all of these skills and knowledge about genomic technologies and all of this infrastructure. So again, on behalf of the organizing committee and the African Biogenome Project, I want to thank you for participating in the one workshop. See you tomorrow again at the same time. I want to say bye-bye at this time.